cancer. But of course, it showed some very interesting signals that how to form the basis of the company Capstan. But of course, there's numerous questions, important questions left to be answered. So the first one would be, how about the on and off target delivery of the CAR by LNP vehicle? They examined the delivery into the T cells. How about to the other organs, other types of cells, which is essential for their safety? And associated question would be, how could you dose the animal? How far can you go? What's the maximum tolerated dose in the mouse? And if you want to go human, of course, the non-human primates. And the important thing is would be to determine the persistency and potency of the in vivo CAR T, targeting B cells lineage markers for the treatment of autoimmune and hematologic cancers. And let's go to the next one. So that will come to their second science paper that came out very recently, which is showed us the promise of their platform technology in the area people most talked about, autoimmune disease and B-cell associated cancers. So first, they show their first generation platform, one that showed in the previous literature in across different organs, as you can see on the right. And not surprisingly, we know LNP goes to the liver. And on the second column, if you can see, that's the one they used in their previous literature. Even more in the spleen, you see tongue condensation in the liver, which theoretically, maybe not directly, but theoretically is a potential risk. So they optimized on the LNP front, and with their lipid 829, which is ionizable lipid, with this formulation, they greatly improved the liver affinity of the LNP, which is very condensed in the liver with this formulation. So they have made great progress on decreasing the unnecessary condensation in the liver. So that's one important improvement they have made with their formulation. So that's the lipid 829. And next slide. They showed, and also another one, remembering the previous literature, they target CD5, which is present in all T cells. And they must have observed some concerning cytokine release signals in their trials. So in their preclinical efforts, so they want to improve on that front. And they have a theory that CD4 is the main contributor for the cytokine release syndrome. Can I reduce that associated risk by targeting CD8 only? And we know for CAR T, of course, their theory CD8, CD4 together make them last longer, but CD8 is the key effector, especially for the cytotoxic function. So instead of CD5 targeting, they choose CD8 targeting this time. And similarly, we could see within the CD8 T cells, we can see CAR expression peaked at the first week. I think this is the x-axis hours, or I forget it's hours or days. Anyway, but that's the CAR expression within the CD8 T cells. And next slide. Next slide, you can see, so on the left, they show what those CAR Ts are doing. They're depleting B cells, and they're working incredibly fast. Within 3 days, they basically eliminate all B cells. And on the bottom, you can see the B cells are effectively eliminated basically within the first few days. Yet, regardless, the 3 repetitive dosing, the last dosing should be on day 6, but on day 7, the B cells are already bouncing back. So yeah, the B cells are bouncing much faster than what we observed with allogeneic or autologous or bispecific treatment. It can achieve B cell elimination, but the B cells are bouncing fast, regardless of repetitive dosing. On the right side, they even increased the dosing to 5 doses, lasted till day 14. And at higher dose, it was effective in eliminating all the cancer and therefore achieve long-term remission in the higher dose group. And next slide. This is the non-human primate. So they really spend some serious money on preclinical testing, which I highly respect and I admire. So this is various dosage groups in non-human primates. So here they show beyond the zero point 0.5 milligram per kg, they are able to achieve long-term B cell elimination even after with three dosage basically. And on the bottom, they also examine the B cell profiles before and after B cell reconstitution. Of course, the B cells eventually come back around 1 month, and they show a more naive B cell profile after reconstitution. But that doesn't really correlate with the clinical, since those non-human primates are not disease model, they're just normal primates. But still, it shows it's a more juvenile, more juvenated, and more immature B cell population as we expected. So let's go to the last slide. So with all the previous introduction, yes.
Can I have a question? So, like when B cell coming back, if they keep dosing, can they also deplete the B cells as good as first time?
So that's the interesting fact. If you look really carefully, it
It
seems
didn't
only
show.
the first dosing. No, they show multiple dosing. Um, for
I'm
example,
saying,
on the point one meg, it seems the B cells are coming back, and with the second dosing, it comes down again. But in general, the following dosing are not as dramatic and effective as the first dosing. First dosing is really dramatic. It's basically eliminated everything within
that
one
could be
day.
incremental. That could be incremental. More important is like, for example, after 1 month, right? They came back
I
very.
I don't know. First, I'm really confident by 1 month there's no CAR T left. The CAR T basically is there just 2 to 3 days at
Yeah, I
most.
understand. But if
Yeah,
these are coming
and
back,
the reason why I don't believe is also incremental, but rather decremental is if you go to the previous slide is really clear. So if you look on the bottom, the last dosing would be on day 6. Yet the B cells are bouncing back on day 7. So if the dosing are as effective as the first dosing, which eliminates from that high level to baseline level within 1 day, and the third dose is really pathetic. So I don't know why, but it seems that the effect of the dosing is decreasing really fast.
okay, all right.
It's just judging from that data. Yeah. Okay. So I'm sorry for taking too much time, but here's the summary slides for the company capstone. So it starts with some really weird data, really great seed and Series A found, really large round, almost top close to 200 million US dollars at data I personally not feel so confident about. But with the recent science paper, they have really made a tremendous progress and advancement that I highly respect. So I think they are moving towards the right direction. But as you could tell, there's no signal about it's overwhelmingly successful than other class. I think it's just an interesting direction that worth exploration and most certainly a lot of modification and improvement. But I personally admire the efforts this company, serious efforts this company have spent, and I think they are moving towards the right direction. And I'm excited to see what's coming next. So that's my share for today. Any questions from the audience?
Yeah. Quick question on the safety. Do you have any comments? But because they just enter phase 1, just dosed their first
Yeah,
patient.
the safety we know from the preclinical testing, they have one serious adverse events in one monkey at their second highest dose. That monkey have the HLH, the hemophagocytic lymphohistiocytosis syndrome. Yeah, so they have to euthanize the animal. It's like the worst side effects you can have with CAR T. Other CAR T product have this one as well, but I think it just speaks of the cytokine release syndrome. It's not direct cytokine release syndrome, but it is a syndrome that is secondary to immune inflammation. So that's what I'm saying. With such large amount of foreign LNP or viral vector, even more flooding into your intravenous system, I personally think CRS and CRS-related other immune risks should be a concern, much higher than other formalities, for example, especially for allogeneic CAR-T. Yeah.
Okay, thanks.
Anyway,
Next.
so I think we're significant delay. Thank you so much, Ian. Very great. It's great.
Thank you.
So the second speaker, we are happy to have Xiangfei, right?
Yeah.
So yeah, can you start? This is a very interesting topic.
Thanks, Dr. Wang. First, huge thanks to Ethan for giving me this opportunity to join you today. 안녕하세요, I'm Fei Xiang from Curtin University. I'm a new kid from this Biospark community, and I can't wait to share this mind-blowing science paper with you all. So, for many years, scientists thought the protein called cerebrum, basically the cell's trash tagger, was very picky. It could only grab about 50 proteins because they needed a special handle called the glycine loop, G loop. So the researcher get clever. They use the computer AI tools to scan every single human protein, about 20,000 of them, and they find something amazing. The first discovery, actually 1,400 plus proteins have the classic G loop handle. That's 30 times more than we thought. Second, 184 more proteins use a completely different handle that just work as well, like using Apple Pay instead of cash, different without same result. The third discovery is some proteins like BAV1 have no handle at all, but can still trick the cerebrum. They are basically master artists. So we just went from 50 to over 1,600 possible drug targets. The takeaway is just Monte Rosa team prove that impossible targets might not be so impossible after all. So next slide, please. But before we see the Monte Rosa breakthrough, I want to introduce 4 scientists who started the whole field. The first are the glue master Stashuler. Back in 1991, the visionary scientist just named this concept molecular glue. He's thinking one drop of small super glue can stick any disease protein onto the cell's ultimate garbage disposal unit, cerebrum, sending villains straight to their molecular doom. Next, the clinical champion, Benjamin Ebert. In 2014, his key paper showed that lenalidomide, already fighting blood cancer villains, was secretly a molecular glue hero all alone. It gluing cancer proteins to cerebrum and crushing them. He showed the world that molecular glue therapy is real. Enter the architecture Ray Deshay. In 2001, he get this ultimate weapon idea, PROTAC, a two-ended molecule boundary cord of justice. One end locks onto the villain protein, the other onto the cell's destruction squadron. The concept is pure genius. The execution, this complex hero, proves breakthroughs. From 2004 to 2010, he transformed a Ray Deshay brilliant blueprint into a working weapon of mass protein destruction, fine-tuning the linkology, every molecule connection until the boundary cord can drag any target protein straight into the cell shredder. Like this fantastic four, each hero brought a unique power, glue, proof, concept, tool. So together, they transform undruggable into druggable. They unlocked the door. Monte Rosa smashed wide open, expanding from a few dozen to thousands of targets. Let's see how they do this. Next slide, please. So Monte Rosa transformed cerebrum drug discovery by systematically scanning the entire human proteome using the CK1 alpha G loop as a searching template. Their algorithm mined
June, it should be the previous slide.
Yeah, so it's a right. I think it's. Oh, okay. The scan Google one. Next slide, I think.
This one, right?
Sorry.
Yes, it should be this one.
This slide or before?
Yeah, this one. Once if
Okay.
we can scan the Google Maps. Thank you. Okay. The team just discovered this 1,424 targetable proteins. The 600 is zinc finger proteins, it's much more than we thought. And 729 proteins is completely different families. Like the panel D, they break this 700 new family proteins to catalogs. There are 96 protein classes and 272 structure domains, including kinases, motor proteins, chaperones, RNA metabolite factors, and more. In short, G loops aren't stuck in one tiny corner of biology. They are scattered everywhere, giving chemistry, like me, dozens of fresh targets family to explore. The bottom line is that's 30 times more opportunity for new medicines. Next slide. Okay, yes, this slide. And finding these proteins computationally, the question is, do they actually work in the living cells? So they designed a clever experiment, thinking giving cerebrum a spray paint gun that tags anything they get close. This is how it works. They attach a biotin tagging enzyme called the tuber ID to cerebrum. When they had a molecular glue compound, any protein gets recruited to cerebrum, gets spray painted with biotin. Then they can fish out all the tagged protein and see what actually binds. Look at these volcano plots, show the results. The higher and the further right a protein appears, the more confidently it was recruited to cerebrum. So the high left to right is a potential target. So the great news is it worked. They find the targets like CK1 alpha, GSPT1, the validation they are made out. But more importantly, they discovered brand new targets like NEK7. So bottom line is they are just not computer prediction anymore. This is a real biology happening in living cells, proving that AI approach may be generously expand what's druggable. But the proximity isn't everything. Let's see which targets can actually be degraded. Next slide. Okay. Thank you. This is G-loop validation. Figure F2H shows the final proof of G-loop prediction are correct. While G-loop sequence vary between proteins, the critical, the big G, glycine, always there. The binding curves on the right are smoking gun. White type proteins, very strong dose-dependent binding to cerebrum, but one with mutation that crucial glycine to asparagine, binding completely disappeared. This confirmed computational rule, no glycine, no cerebrum binding. They tested this across multiple targets, proteins, and compounds, same results every time. The algorithm works, and we understand the molecule mechanism. Next slide, please. Okay, extending this is where things getting really exciting. The team moved beyond kinases to other proteins, completely different function, like RNA processing, gene regulation, metabolism, you name it. The panel B shows the binding results. 8 out of 11 worked beautifully. The red curve shows strong dose-dependent binding and disappeared when the mutation the G-loop glycine. But there is a real reliability check. The panel C shows the binding doesn't automatically means degradation. Most proteins binding cerebrum still don't get degraded with this particular compound. This tells us we are successfully expanded the target space, but we need different chemical approach for each protein family. The sequence analysis confirms that only glycine is truly required. The rest can vary widely. So we have proven that 1,400 plus targets are real, but turning them into medicines will require a lot of more chemistry work. Yes, next slide. Next. Discovery of helical. Yes, this slide. This where is discovery gets even more exciting because the team asked, what if we focus only on the essential G loop that actually contact cerebrum? So they use minimal templates. They finding 184 proteins with helical G loops instead of beta hairpin G loops. This is a completely different structure type. The example is mTOR, very famous major cancer targets that's been incredibly difficult to drug. Look at panel E. This is a crystal structure shows mTOR's helical G loop making identical hydrogen binding with cerebrum. just like beta-hairpin variants. Same chemistry, different shape. The panel K shows the works for multiple kinases. This discovery essentially doubles target space. We now have both beta-hairpin and helical G loops working through the same mechanism. It's like finding out both right-handed and left-handed people can shake hands equally well. Okay, next. Why each target is custom chemistry. Yeah, thank you. Okay, this slide reviews the hardest part of drug discovery. Binding doesn't equal to different. Look at next 7. Compound 1 can result in cerebrum perfectly, but it doesn't get degraded. Only when they find compound 5 with the right chemistry did the next 7 actually get destroyed. The crystal structure show why. Different compounds create different protein-protein interface. Panel K shows the footprints were completely different between NEX7 and CK1 alpha, even though both have G loops. The validation experiment proves NEX7 needs extended contact beyond trusted G loop. So while they proven that 1,600 plus proteins can be potentially targeted, each one will need its own optimized molecule glue chemistry. So it's having 1,600 different locks. You have proven they can all be opened, but you need 1,600 more different keys. Okay, next. Beyond the G loops, the wave 1 surface limited groups. Next slide. Oh. Can I go to next slide? Okay, thank you. This slide is truly revolutionary. The team asked, what if proteins doesn't actually lead to G loops at all? Just need to look like G loops on their surface. They took an example, BAV1, a major autoimmune target that has 0 G loops, and used surface matching algorithms to see if any part could mimic non-cerebrum binding surface. Amazingly, they found that wave-1 SH3C domain has a surface patch that looks just like GSPT1's G-loop interface. So same chemistry, same statics, but completely different underlying structure. It's molecule mimicry at its finest. The breaking through shatters the G-loop paradigm entirely. So if proteins can fake the right surface features, potentially thousands more targets becoming accessible beyond even 1,600 G-loop proteins. Drug hunters have gone from struct-based targeting to surface-based targeting, a completely new way to thinking about molecule glue. Next slide. Discussion. So this slide beautifully illustrated why Cerebrum is such a powerful platform. Panel A shows the PPI hotspot, red region, where all targets make contact. It spans 2 flexible domains, giving Cerebrum incredible adaptability. Panel B, like a gallery of molecule footprints, showing how each target uses this hotspot differently. Notice the last picture, the VAV1's footprint looks completely different from G-loop targets, yet they all use the same core hotspot. This explains why each target needs custom chemistry. They are stepping on the same dance floor, but doing completely different dances. This gives us a roadmap for rational molecule glue design. Understand the footprint, design the chemistry. So we've gone from accidentally discoveries to predictable platform engineering. Thank you. Last slide, yeah. Next. So actually, last slide is talking about the drug discovery has always been hard. AI, our CADD, give us new tools, but it doesn't change the fundamental challenges. So here's what I believe. Every experiment that fails teaches us something. Every model that's wrong points us toward what's right. Every patient waiting reminds us why we do this work. So keep building, keep trying, keep hunting. The next breakthrough could come from anyone in this Biospark Zoom meeting. Thank you. 감사합니다. I'd love to hear your thoughts and questions. That's all.
Yeah, that's a great presentation. I think every slide was very
Yeah.
carefully presented and organized. I really thank you so much. And also I can feel your passion and also
Welcome.
your insight. Thank you so much. So any comments from the audience and also, everyone?
Yeah, well, quick question for Sean. Obviously, one thing you point out, binding does not automatically leading to degradation. Do you
Yeah.
feel like majority, although this works nicely, expanded the binding partner for cerebral, but what's the next computational kind of scoring, can they say, is leading to the higher probability of real degradation? Or you have to do
I see.
experiment to prove degradation?
Very good question. Personally experiments, I feel something. It's like so many AI companies, not as well as Monte Rosa or other company, because usually some experiments prove something, and after that they're using the model or computer to build some model to explain the situation. So sometimes it's not come first, actually. Personally, I think especially for this kind of work, if you ask me the limitation of their AI changing is they just focus on the how to the rigid form of the surface of proteins. But I see some like Schrödinger or they have the dynamic molecular dynamic more how to say more FVP calculation, more powerful to detection one or two special protein situation. So maybe it's still need, yes, the wet lab, the experiment always the key, and then maybe 90% work should be done by the wet team. But AI, of course, help us, give us a lot more chance.
Okay.
So Xiang is the first program VAV1 you mentioned. This is very surface-oriented drug discovery. Is this the one the highlighted and the molecule was purely computational designed?
I don't think so. I have to check, but as I know, this is only one example in the pipeline, but it's maybe going to clinical trial. But usually, if you're talking about computer design, actually, it's not always 100% or 20%. Like in our lab, we don't use computer design anymore in 10 years ago. But now my professor asked me go to his office, open the Schrödinger 3D picture, and he give me some red and blue point. He say adding something here, and I docking. Oh, amazingly, it's increased very very good. But I also can dock in like 10,000 in naming libraries, get 5 or 10 compounds, seems work better. But the problem always how to silence it. So who can make it faster, and the chemistry, the problem challenge you can finish early, or with like Wuxi APP's help, you can make it real. It's more important. So if you think the computer, maybe most of the team using more and more, but usually it's 50% or 20%, but it helps, but not all the work by you can design automatically for one program, something like that. Yeah.
I see.
Feedback.
Thank you so much.
Yeah. Thank you.
Right, thank you so much. Given time, we have to move forward for second topic. Thank you so much, Sean, and I wish you were attend our events more. Thank you.
Yeah.
All right, so given time, let's move to the second section, oncology and autoimmune disease. So I will start to talk about the first topic on this section. Merck acquired Verona. So this is a company founded 20 years ago in UK, and then they have been focused on biology, though they have been focused on PD inhibitors. I think 2 years ago, I think Ethan mentioned about this company. They have some early clinical trial data. So they have been focused on PD3 and 4, and they made a small molecules as a dual
I think
inhibitor.
I have also shared this company on our monthly news not so long ago, a year ago, probably.
Either Ethan or you, we will talk about this.
Yeah, I'm positive it was me.
Yeah, okay, that's great. So anyway, so they spent about 20 years for to build this company, but only until 2 years ago, I think they have seen some very exciting data. But before that, the company almost died. So last year, the first program on COPD has been proved from FDA. It's basically is a first in class COPD drug, chronic obstructive pulmonary disease. And this is the first class of drug approved in 2 decades. And then very quickly, recently this month, Merck announced potential should be there acquisition with $10 billion. This is a very successful stories. So we can dissect this company and programs step by step. So this drug actually is oral, no, inhaled is a small molecule, but it's inhaled. First inhaled COPD treatment. So they can do 2 jobs. Okay, one is called a bronchial dilation. It's basically the lung function and also anti-inflammatory function. So this is dual inhibitor, but targeting 2 different pathways. So why they want to do that? So first of all, the pathophysiology of COPD has huge unmet medical needs because it involves airway narrowing because lung, need to inhale, exhale airs. So that's why the bronchial constriction it can be very bad to affecting the life quality. And also this is basically about the smooth muscles contraction in the lung. And the second is inflammation. Chronic inflammation in the lung with both innate and adaptive responses will affect lung function, especially mucus overproduction. That will take the space in the lung. So that's why when you inhale and exhale, the lung volume, the air volume can be different, basically basic lung functions. And then this is also chronic. So the standard treatment, as I show here, the cause could be damaged, the lung and also the airway. So basically there are 2 types, but people normally talk about 3 types, but I think in general it's 2 types. One is about lung function, is more focused on bronchial dilator, either from LAMA or LABA, just like different classes of drugs, but the goal still is bronchial dilator, is more like lung function. So for example, like LABA is more about the beta adrenal receptor agonist, and LAMA is more like long-lasting, more about the muscarinic agent is more like different type of modulation of the lung function. And another way is inflammation, how to do anti-inflammation. So for example, steroid. So all of those has been proved and the market has been increasing, and people also think about how to combine with each other. But unfortunately, they have limited efficacy and also have some resistance problems, especially for moderate to severe disease. So as I mentioned, this is a huge the disease with huge unmet medical needs. As you know, steroid have some side effects and also cannot be chronic. So that's a major problem for chronic disease associated with autoimmune or inflammatory disease. So why they are talking about PD3 or 4? Because PD3 is more on smooth muscle, especially in the airway. So, if you preclinical study talking about lock it out or inhibitor can make the lung, the function be improved, especially bronchial dilation, because this is a drug. This is a major molecules control CAMP or CGMP. But the PD4 is more inflammation, or more on neutrophil, macrophage, and T cells more. That's why if you target both, you might have dual function to target the majority of the pathophysiological factors related to COPD. So that's why they have been focused on these small molecules, and it's a novel selective inhibitor, can block both 3 and 4. So with this, you can control smooth muscle and immune cell and potentially epithelial cell around the non-disease lesion. And then with this mechanism, you especially control mechanistically those 2 enzymes to control CGMP or CAMP, which is important for those functions, especially for promoting immune responses, contraction. So that's why with this mechanism, you can control both lung function, bronchial dilation, as I mentioned, that's number 1. And the second is the inflammatory responses can be controlled, can be reduced with the PD4 inhibition. And another way is secretory function is more like mucus building up. That kind of function can be also controlled by epithelium or or the basic lung function. So that's why this is a very interesting ideas with one drug to control two different pathways, but which are synergistic. So in oncology, we are also talking about synergy. But I think for this disease, especially for this case, this is really interesting pathways and targeting major mechanisms for the disease, and they have a solution to target both, and in a synergistic way. So PD4 is many immune cells, and the PD3, some of those can be immune cell as well, but some, they synergistically control different subsets, especially PD4 and the PD3 can be also on small muscle, and the PD3 also in the epithelium. So if you target one of those, it's not very enough, but if you target both, then you will be control the mechanisms as I mentioned. So this is the first novel inhaled mechanisms, and like they basically important for CGMP or CMP breakdown, and important for these three directions as I mentioned. In order to reduce the whole body exposure and the toxicities, they also make this drug the first inhaled drug for COPD. So that is also the quite novel. They didn't think about the systemic oral or other approaches and think about, because this is a lung, very straightforward. So you use inhaled. So the data, normally when people talk about COPD is about the volume of air. When you forcibly inhale and exhale, you can measure the volume of air. That is a measurement. So they have done 2 trials with about 700 people, and in each trial they have seen very similar data. The volume of air can be greatly enhanced, and also 33% or 43% of reduction for the exacerbation rate. It's basically to measure lung function, and is greatly improved. So based on those type of data, as I mentioned, especially those type of rate of reduction, which reflecting the lung function, this drug was approved last year. But this company is still going beyond, to test other indications. For example, lung cystic fibrosis, and bronchial, tysis. So this is also related to lung function plus inflammation. The trial is still ongoing because you want to target both inflammation and also the lung function at the same time. So hopefully this drug for other indications will be successful as well. So this is other programs. The first program has been approved. They're also working on other disease, interestingly asthma, right? Systemic fibrosis and others. So based on all this information, another information why Merck want to acquire this company, because the sale. So this year, the fourth quarter, the sales of this drug is already $72 million, the first quarter. This is just like 1 or 2 years after, get approval by FDA. So over time, you might get a much more sales, become a potential blockbuster. That is another reason why Merck want to acquire this company. So that's why I think, this drug is focusing on very interesting, very potent molecules, control CGMP, CMP, and also very innovative drug design, and indication, actually more than one indication. Several indications have huge unmet medical needs, and also persistence, and also their focus of the science, and also interesting drug design, leading to the success of Verona. But beyond PD3 and 4, there are actually more than several other targets which already have some success. So this is data I already showed. Actually, there is another drug targeting PD5. The most famous drug is called Vigro, that many people should be familiar with. So PD5 actually controls CGMP only, but it's more on vascular smooth muscle compared to PD3 is more on airway smooth muscle. So that's why in the beginning, when people develop PD5 inhibitors, people were more thinking about heart disease, but unexpectedly, unexpectedly, they found this drug, because control vascular smooth muscle, can actually control, like, for male, sex function. That's why they made the drug Viagra. So this is a very interesting drug history. If you are interested, you can look at how PDE5 inhibitors originally they wanted to develop for other disease, but eventually become a very popular drug for male. And of course, they have some side effect. But quite interestingly, these PDE5 inhibitors recently was found to have other function even beyond the male sex function. For example, this is a mechanism for how Viagra works. So basically, sexual stimulation, and then you have some GDP building up, and then you will control the smooth muscle, especially artery smooth muscle. This is important for sex function, especially for male. But this pathway is not just like smooth muscle. So recently, there's a paper published by one of my friends in Westlake University in Nature. So what they found is PD5 actually expressed on dendritic cells. So when dendritic cell migrate from tumor to draining lymph node, they found PD5 is one of the gene greatly modulated, highly induced. And because this suppression, they use a very comprehensive screening system to screen how the dendritic cell coming from tumors to the draining lymph node. And also they have two different screening, quite comprehensive. And in both screening, they found PD5 is one of the most extensive abrogated genes, from when the DC coming from tumor to the draining lymph node. So they found actually if they inhibit this gene, the tumor draining lymph node, you will see much more DCs. Okay, so this is an important function that one of the molecules specifically control DC coming out from tumor to draining lymph node can be important for T cell function, DC function. And that's why I think it's quite interesting. They also show some data inhibitor also can be important, be synergistic to PD-1 inhibition, can be important for cancer immunotherapy. So interestingly, another paper recently also talking about PD-5 inhibition beyond beyond DCs, also control myeloid cells. So that's why I just want to share this story beyond PD3 and 4 and also PD5. So all this type of drug discovery, the history, and also those stories will provide insight how those drugs been developed and how the company been successful, and how we can see and identify more mechanisms and more potential therapeutics can be useful for the disease.
So thanks, Jun. I'm sorry. I just one point. So I would like to bring up that in addition to the PD4/5 pathway, another one to be noticed would be the type 2 interferons, and Dupixent has been accumulating some really promising results. I would say although it's similar, but worth noting would be the control groups. A very important point for the Verona trial, the majority of the patients have received only Lama-Lada combinations. Yet for the Dupixent, although the response rate seems to be similar, but the majority of the patients have received the tri combo of the Lama-Lada plus ICS inhalable corticoid. So I would say the landscape is not only focusing on making a better, safer PDE5 inhibitor, but also in the modern context of multiple biologics, especially with the type 2 interferons, and how to remain competitive on this front. But I really like this one because this is an exhaler. This is a nebulizer. So I think that's very convenient and natural for arthritis patient and asthma patients. Okay, that's all my comments. I'm going to mute now.
So thank you. And I think for the landscape for this company, they have several trials ongoing, and asthma, cystic fibrosis, and bronchial how to pronounce taxis, and then all those type of stuff. I think the inhale and also the drug mechanisms and some of the trial design, they are also thinking about some potential combination with existing drugs. All these are interesting, and because this is unmet needs, so they have pretty much space to go forward. So I think to summarize, I think this story tells us we should focus on unmet medical needs. You have space, and also based on science, and this is the potent. They are focused on potent targets, and innovation. Oh, sorry, those targets, as I said, both PD3, 4, 5, they have important biology controlling different location. For example, PD5 control, the smooth muscle in different location, and great biology. And also you have to be innovative on drug designs and even think about delivery approach. And they have more than 2 decades of determination, and they are visionary and have great patience to finalize their product. And also biology is so interesting. That is what I can say. So yeah, with that, so if you have any questions, we can discuss a little bit before moving to other topic. All right, so maybe we can start with the second topic, and then we can always go back if any further questions. So Hongyu, thank you.
Hi, good evening and good morning. So today I'll be presenting the recent ODAC opinions on the ADC drug by GSK. So this is a BCMA MMAF ADC called Blenrep. So this is the breakdown of my talk today. So I will first discuss the voting results and voting questions. And then I'll show you the trial data for these applications and the FDA's position. The second half of the talk, I'll be discussing mostly what's behind the voting questions, why they voted this way, and the drug itself. Next slide, please. Yeah, apparently I misunderstood the format. I should put the title on the top. But anyhow, so these are were the voting questions and the results. So this is Blenrep as a component in a combination of Blenrep, Velcade, and dexamethasone, so BVD, or Blenrep, pomalidomide, and dexamethasone. So this is in the second-line setting for multiple myeloma. So the question, the exact question was put on the top of the figure. So this is the question whether in the proposed dosage in the proposed population, the compound showed favorable benefit and risk ratio. So as a BVD, so there's a 3 supports, 5 rejections. And then as the BPD combination, so there's only 1 yes and 7 no's. So in addition to the answer to these asked questions, there are some concerns regarding there are not enough U.S. patients being enrolled in these trials. And also the Black and senior people older than 75 years old were also underrepresented. So it's very noticeable. The question the FDA asked was not if the drug should be approved. What rather is mostly on the dosage. So I'll come back to this point later. Next slide. So this is the trial data. So as you can see in both trials, as a BVD or BPD, the compound showed very good efficacy. In the BVD in the DREAM7 on the left, the BVD improved almost 2 years of PFS over the control arm DVD. This trial also hit the overall survival endpoint. The second trial on the right, DREAM8, as a BPD, it also improved the PFS quite dramatically. The control arm was 13 months. The median PFS for the drug treated arm did not achieve. This trial did not hit the OS endpoint, probably because this is a much smaller trial. So there are only 300 patients in this trial versus first one had over 500. So this is a smaller trial. It didn't hit the OS. This is probably why they got fewer supporting votes for this combination. Next slide. So although the efficacy was fairly dramatic, there are some safety concerns. As you can see on this page, over 90% of patients showed some ocular tox. Close to 80% had a grade 3 and worse. So events included blurred vision, keratopathy, and ulcers. As many of these side effects resulted in a dose interruption, dose reduction, and some termination. So on the right, the two tables on the top, that's a breakdown of the tox. You can see grade 3 and 4 toxicities were very common. The bottom table showed the visual acuity. Side effects, that's the functional side of the ocular tox. You can see over 60%, maybe you cannot see, but I can tell you over 60% of the patients had a vision drop to 20/50. A little bit over 10% drop to 2200. So on the left corner, that is a simulation of this vision drop. As you can see, 2200 is really bad. In the US, that is considered legally blind. So it's about 10% people, the best corrected vision is only 2200. So these are not trivial talks. So next. Yeah, so different agencies had a different position and made a different decisions on these agents as a combo. Incidentally, the compound as a single agent was approved in 2020 through the accelerated approval pathway at the same dose used in these two trials, 2.5 mpk, in the fourth line setting and beyond. So in that setting as a single agent, it showed the 32% response rate and a PFS of 3 months. It shows pretty severe ocular tox, but it was not as bad as observed in the most recent two trials. So as a combo in the second line multiple myeloma, because the advisory committee's negative voting results, FDA postponed the PDUFA date from July to October this year. So FDA may still approve this. It may count the physicians adjust the dose in practice, but it may also ask GSK to do a smaller trial using lower doses. Other agencies made a different decision. So very recently, this week actually, there's 2 more approval. So UK, Canada, EU, and Japan all approved this drug as a combo in the second line setting, and NICE actually endorsed it. So this is not very this is not trivial. So it's again, the endorsement from NICE is not easy to do. So the FDA's position basically it was more about the dosage. So the FDA believed at the lower dose, the compound might show more balanced benefit and risk ratio. So this is where things. I think this is what where debate is on now. So next slide. So this ODAC meeting highlighted the critical role of dose optimization in oncology drug development and drug development in general. So historically, if a cancer drug that can improve the OS, they would have been a slam dunk with FDA. However, since FDA rolled out the Project Optimus in 2021, so they emphasize more on optimizing both efficacy and safety, not just efficacy alone. So that actually shifted from the old MTD approach to the new OBD, which set for optimal biological dose. So they want to have a balanced efficacy and safety. So this is probably why the question was asked at the proposed dosage, and that question, some experts believe, framed the voting committee toward more to whether the dose is optimal than whether the drug itself is approvable. So FDA suggested the drug could have a more balanced risk and benefit ratio at lower doses, as these ocular tox were clearly dose dependent. And they say the GSK might have missed some opportunities in phase 1 in finding the optimal dose. So this is not only, this dose optimization is not only critical in oncology drug development, it's in drug development generally, especially for drugs with narrow therapeutic window. So the GLP drugs is probably the best case for this. The GLP-1 drug on high doses were not very well tolerated, but through a dose titration scheme, the side effects can be reasonably well managed, and these drugs become just a monster drug now. Even DEX itself has gone through clinical trials to show the dose reduction can improve the risk-benefit ratio. So we'll see whether the FDA will approve this drug and rely on physicians to modify the dose, or they will ask the sponsor to actually prove this in clinical trials. Next slide, please. So that's Project Optimus is only one factor. There's another reason why the committee voted this way. Multiple myeloma actually in the past 20 years has quickly become a very crowded space. There were at least 19 new drugs approved in the past 20 years, including some BCMA drugs, including CAR-T and bispecifics. So on the right, that's a chart provided by the FDA. So in the second-line setting, you can see there are multiple choices, including the drug combo used in these DREAM 7 and 8 trials, but also there are CAR-T and bispecifics. So when you have multiple alternatives, your leverage is not as strong. So this is in comparison with another drug that made news recently, that's the DMD gene therapy drug, Elevidys. So this drug has the questionable efficacy, to say the least. It never met a primary endpoint in clinical trials, but it got approved. Actually, when FDA asked the sponsor to halt shipment of this drug this past week, there is a strong public uproar against that. And actually, some people suggest Vinay Prasad, which is the director of this section in FDA, should resign because of this. So that's the leverage you have when you are the only option in a deadly disease. But when you have combinations that make it harder. Next slide. So more fundamentally, so this is also this results, this voting results also highlighted the limitation of distribution-driven drug design. So this is more fundamental in the basic science of ADC. So eye is a very special tissue in terms of drug distribution. Is considered a sanctuary site. It has several barriers like blood aqueous barrier, blood retinal barrier, to keep the drug from getting in, and also has a pretty high content, high mucin contents to limit the drug diffusion. However, if the drug find a way to get into eye is a fairly vulnerable tissue because the corneal cells have a limited regenerative capacity. It has very high metabolic demand. It's easily interrupted by chemicals. And eye in general is just very, very delicate tissue. So this is similar for the football fans. It's similar to a football game. The quarterback, the reason the quarterback is not sacked constantly is because the offensive line protects him very well. Once the pocket is broken, the quarterback is actually very vulnerable. So BCMA is not a random target. It's a very well validated target. So there's multiple CAR-T and bispecific against it get approved. But ADC still carries some unique risks. The most fundamental problem for ADC is there's always some baseline non-specific catabolism, and the payload get released in healthy tissues. And pharmacokinetically, ADC cannot get excreted until it's catabolized to get payload released. So that also posed a constant danger. And ADC normally is released intracellularly for MMAF. This payload is very poorly permeable, so it's going to stay in cell for a long time to cause damage. So this is another problem for ADC. If this compound find a way through non-specific endocytosis to the corneal cells, the MMAF will have a hard time to escape. And there's another possibility, maybe that the corneal tissue is just very intrinsically sensitive to MMAF. So that's a possibility too. Next slide. So actually, eye is very sensitive to drugs, not only to chemotherapy, even some benign drugs can cause eye tox. So these are just two recent studies, one on the dry eye risk and one on the keratitis risk for commonly used drugs. So you can see a lot of drugs can cause that, including Viagra, June just mentioned, actually can cause visual tox. But you can see on the top in both charts, that's our drugs approved for eye disease. So they probably optimized to get into eye. You can see once the compound get into eye, even though they were approved for the eye diseases, they can cause much bigger problems for the eye. There's one compound, I don't know if you can see it, paclitaxel. So that is a tubulin inhibitor like MMAF. So that can definitely cause eye problems. Next slide. So in summary, Blenrep is a pretty interesting drug as a single agent. It was in a later line setting. It was actually not efficacious enough to beat the control arm. It was pulled from the market not because toxicity, but because lack of efficacy. As a combo, as shown in the DREAM 7 and 8, they were very efficacious at high dose, but it showed, unfortunately, pretty severe ocular tox. So in addition to that, the combo need to compete with other regimens, to make it harder for the FDA to actually just prove based on efficacy. And the Project Optimus specifically asked the sponsors to optimize dosing in the early trials, so they can have a more clear answer on the optimal dosage. This also shows manipulating drug distribution in drug design is a tricky business. Actually, all targeted delivery systems have some built-in bugs. You just have to deal with them individually. However, all is not lost. The compound already got approved in this setting as this combo in multiple major markets, and FDA probably still will prove this. The fact they actually delayed the PDUFA date depends on how you look at it, is probably a good sign. So that's what I have to say today. If you have any questions, I'd be happy to answer.
That's great, Hongyi. Any comments from everyone?
没有 comment 有一个问题啊，就是它这个药这么的，就是怎么说呢 ，toxicity profile 不是很好的话，它虽然被 approved 了，它现在的这个 commercial 的情况怎么样，卖的如何？
这个都是刚刚 approved， 那个 UK 是6月份大概 ，Canada EU 是这个礼拜 ，Japan 我忘了什么时候了，它可能现在还是 early stage， but you know the oncologists, at least some oncologists want this compound to be approved, is that they know how to optimize the dose, and they say most of multiple myeloma drugs were not optimized for dose. So they figure out how to use it anyway. So I think some oncology at least lack the efficacy, and they think they can manage the side effects if the drug is on the market. But you know, FDA wanted sponsor to do this proactively. They wanted to get optimal dose. So you know, I think if it wasn't for the Project Optimus, it probably should have been approved. Just you know, the physicians are very experienced in optimizing the dose in practice. But because of this, they probably, yeah, I don't know. This is maybe against their. If everybody get exception, the project wouldn't have much power. That's just my take.
So there's a question from
谢谢红宇。
There's a question from Sichuan. So given the severe and dose-dependent ocular toxicities observed, has GSK explored or published data alternative dosing and to balance efficacy and toxicities and tolerability more effectively?
Sorry, I didn't get the question. So how would GSK?
Yeah, has GSK explored alternative
dosing? No, I think the FDA. This is just based on the discussion on the internet. I think that people say FDA want them to do a trial with lower doses and see if they can improve the risk-benefit ratio. You probably lose some efficacy, but you probably get more improvement in the safety side. I don't know how big of the trial it would be to prove that. But I think there's still pretty decent chance the FDA just prove it and ask the physicians to find those. Yeah.
That's very
Yeah,
interesting.
maybe just a quick comment. I think there are some small companies that are developing specific treatment just to manage this ocular tox for blend wrap. I think it's a clear there is benefit, right? And it's also clear, it's also clear there's severe ocular tox. The 70-80% grade 3 is really pretty bad. But I think because of the benefit, I agree the drug is likely to be approved by the FDA, and there will be some perhaps post-approval commitment for GSK to do. I think it's tricky because you can manage this issue by, let's say, dose reduction when you have the problem, but the efficacy perhaps would suffer because there's two strategies. One is to give a lower dose to begin with, or you can give a higher dose and then drop the dose after you see the AEs. But the evidence so far seems to suggest the efficacy is better when you give the higher dose at the beginning and then drop the dose afterwards. So let's see how the GSK is going to manage this problem.
Yeah, I think I think you're absolutely right. This is very efficacious with very significant tox. It's just need to be used, properly.
All right, thank you. I think given time, let's move forward. So 丽华。
好，谢谢君。我来讲一下这个news，可能大家不知道有没有注意，就是这个实际上是6月底的一个 news，就是这个 Revolution Medicine 啊。
丽华，sorry, can we stay in English because we have non-Chinese speakers
sorry.
online?
Yeah, thank you for the reminder. Sorry about it. All right. Okay, so I will update this one particular news feed to Revolution Medicine and Summit Therapeutics. So the two molecules, I think for the regular Biospark listeners, you're probably all very, very familiar, so I won't get into them too much at all. It is a collaboration between the Revolution Medicine, which is really leading the development of the RAS inhibitor, especially the uniqueness of their RAS inhibitor is not typical as many of those covalent G12C, rather is using this very unique kind of tri-complex inhibitor to inhibit RAS, and they have a portfolio of RAS inhibitors coming from the first pan-RAS inhibitor. And then, of course, for Summit, everybody's very familiar with the bispecific antibody targeting VEGF and the PD-1. So the reason I want to share this news is for two reasons. One is from a scientific point of view, why this is a interesting news. The other one, I think, is really from a business point of view, that obviously Revolution Medicine is the leading biotech company in terms of developing RAS inhibitor. RAS arguably is probably one of the most important oncology targets or oncogenes. And Summit, we all know in terms of bringing this bispecific antibody, potentially challenging PD-1 inhibitors, right? So it's really in the news in the last couple of years. And if you pay attention to this Revolution Medicine is really leading the clinical development, and Summit is simply supplying the medicine to Revolution Medicine, to RevMed. So next slide. So when people do combination therapy in oncology, typically you try to achieve two things. One is you want to deepen the efficacy or the response that the single agent may not be able to fully achieve. The other one is related, but not necessarily the same, is you want to prevent or delay the emergence of resistance. Most of the targeted single agent oncology compound pretty much always have resistance emerging. So this is the new AACR poster this year during the AACR 2025, published by Revolution Medicine, that we're all very familiar with the RAS inhibitor resistance by the G12C from Mirati or from Amgen before. And this is the first time Revolution actually reporting for their tri-complex inhibitors, what are the resistant mechanisms. And they're coming from both sequencing patient, but also from treating preclinical models until the model become resistance. And now surprisingly, the targets on the same MAP-Kaisen's pathway up and down can contribute to the resistance. And one of also the interesting mechanism is the RAS itself get amplified. So coming back to why a combination making sense is that as much as the Revolution Medicine has shown amazing efficacy, the resistance is still emerging. And so next slide. The other reason I want to really talk
sorry.
about, go
Can I
ahead,
ask
Jun.
So,
Yeah.
so I didn't get the rationale very much. So you mentioned what's a rationale here using upregulation? Which data maybe a little which data, like what's the rationale of
You
the-
mean rationale for combination with PD-1 VEGF bispecific?
Yeah, with RAS.
So this poster is not the rationale for that one. This poster is simply saying resistance really emerging. Revolution does have additional publications where they testings preclinically show that the combination makes sense. The other maybe more straightforward way to look at it is RAS inhibitor or RAS mutations are in, in top three tumors, right? So pancreatic, non-small cell lung, especially lung adeno, and the CRC. And especially for lung, non-small cell lung, and the CRC, that's also where the checkpoint inhibitor has been used. So just simply from having a standard of care, you can combine them too. So this particular poster isn't exactly about why you should support a PD-1 VEGF bispecific combo. This particular poster simply saying there are resistance emerging, so you do need combination.
I see. Okay, thank you.
Okay. So the other one I really want to bring this in is actually given the current kind of biotech environment, right? So the news we tend to hear is people having difficulty raising funds, so they have to really put in all their resource to leading asset clinical development, or maybe like we shared a couple of news even in today that how much the biotech hoping to be acquired by large pharma. And so one thing I found Revolution Medicine to be interesting in this environment is that the different things they have been doing. And I've been asking this particular question is, are they building themselves to be the next biopharmaceutical companies? I know we had a discussion, right? So after Vertex or Regeneron, who else is the next? Or maybe after Alnylam, who else is the next biopharmaceutical company? And this is obviously the simplistic view. Every biopharmaceutical company becomes successful for different reasons. But I think maybe the most important reason is they do need to have a commercially highly successful drug, and that really solving clearly unmet medical needs. And ideally, scientifically, to support this commercially highly successful medicine, it's better to be addressing a very important target for a disease with unmet medical needs. And scientifically, it would be even more interesting if it's a new modality or it's a completely new way of targeting the disease or the compound or the targets. So if we look at this in this angle, look at the Revolution Medicine, let's see what they have. So they are obviously, next slide, targeting extremely important targets in oncology. Oh, the previous one, June. Yeah, RAS. And I think the tri-complex, even though the first inhibitor came in from Warp Drive, and I think if you look at the portfolio they've been developing, they definitely made this into a platform to be able to targeting additional RAS mutation. And we'll share the next slide. I think they want to use this approach to target additional targets. And they have not approved yet. They have a highly commercial successful medicine because they are still in phase 3 trial for pancreatic cancer. And so that's an unanswered question. Then the question become why I'm asking this question, are they positioning themselves to be the next biopharma? So I quoted this particular interesting simple quote, where there's a will, there's a way. So the question is there a will from a Revolution Medicine's point of view? And again, I don't have any insider information. This is just purely from observing from the outside. And the top screenshot is coming from their pipeline or their science on their website. So they are moving beyond RAS. And if you look at their pipeline, they stop all the SHP2 like what they used to have. Their clinical pipeline is only RAS inhibitors, and they are moving beyond the RAS in their discovery pipeline. And they do back that by action. So also in July, there's a news that they are actually collaborating with this AI drug design company called Ambic. It's a California company too, on a research collaboration looking for new drug candidates. And they back this up by finance. So they have to invest certain amount of money with Ambic. So that's very unusual that not only you are doing discovery, you actually are investing even in collaboration. And is there a way? So this question is there money for them to do that? And so again, the end of June, there is announcement between Revolution Medicine and Royalty Pharma. I think our Biospark network have people even working in Royal Pharma. So I will not pretend I know very well how Royal Pharma work. But definitely there are significant amount of money invested for Revolution Medicine to really pushing their RAS inhibitor going through the global clinical development, including commercialization, which is very different than most of the small biopharma. And another news, which is actually old news, is the end of 2023. I don't know how many of us paid attention. Actually, Revolution Medicine at that time acquired EQRx. Again, many of us are very familiar with EQRx. EQRx is probably one of the first US companies actually being extremely active and exclusively getting their asset from China. Unfortunately, because of the regulatory environment that they have to fold their business, but they were able to raise a lot of money. So when they fold their business, they still have about $1 billion in the bank. And the acquisition of Revolution Medicine of EQRs are essentially moving those $1 billion from EQRs to Revolution Medicine to support Revolution Medicine's portfolio. So again, you can see Revolution Medicine is a publicly traded company. You can probably check their finance, but I think just from those news, my reading is they also have financial means to truly fully develop their portfolio, including potential commercialization, if they can get FDA approval. So I found it's kind of a breath of fresh air in this environment. When most people are talking about survival, they are actually, I guess, thriving. So that's all I have for this news.
All right, thank you, Nihua.
Oh, Haoran has a question.
Yes. I don't have questions.
Okay. Anyway, we can go start. We can move forward because I think it's getting a little bit late. So how then can you start the third section and metabolic?
Okay. Yeah. So last Biospark meeting we talked about minerase drug that's very similar as this one, the AstraZeneca's baxdrostat. They all target for the aldosterone production enzymes, which is CYP11B2. As an inhibitor of this enzyme, it could lower the aldosterone level in the body that can contribute to a lot of the uncontrolled hypertension especially in the late stage kidney failure patients. In this case, I believe AstraZeneca have a very competitive molecule with only 5 nanomolar of EC50 comparing to Minera's 9 nanomolar, and both drugs are very selective over the cortisone production or cortisone enzymes. So that makes it very specific to control blood pressure instead of interfering the cortisone production in the body. So but Baxdrostat data is not released. It's just said it's highly selective, but lorundrostat have a over 300-fold more selective over the target. I believe these two drugs probably is having similar selectivity profile. Okay, next slide, please. Okay, so this equity is a acquisition from a small biotech, Syncore. I think it's from AstraZeneca with 1.3 billion payoff. So it's I don't have the phase 3 data yet, but I pull up some phase 2 data, which looks very promising. And with a very a small dose, 0.5 milligram, it could lower the blood pressure by almost like 17 or 17 or 16 millibar. So it's a very significant and clinical meaningful drop in the blood pressure, especially in the late stage patients like kidney failure patients. So you also have a very good safety profile, making it good options for those patients as well. In this trial, it meet all the primary endpoint and the secondary endpoint with 1 or 2 milligram of drug, which is a significantly lower dose than the competitors. Okay. Next slide, please. I have a comparison between these two drugs. Yeah. Okay. So on the left is the AstraZeneca's Baxdrostat, and on the right is Mineralys lorundrostat. And obviously the AstraZeneca's molecule is much smaller, and I think it has more like hydrophobic core structures, which make it the half-life of this much longer than the Mineralys molecules. So it's almost double or triple the elimination half-life, which is crucial for sustained and stable blood pressure control. And it's also very important for late-stage patients in kidney failures. So overall, I think on the efficacy side, they're quite similar. They both reduce the system blood pressure by over 10 mercury. So in that sense, a better profile of the drug toxicity is also crucial. Baxdrostat have slightly higher hyperkalemia problem than the lorundrostat, which has a slightly lower efficacy. So I think that's that can be controlled by like a dietary intake of the potassium and other measures. So overall, in comparison, and I think this is a much even better version of the molecule than the Mineralys, and I have a good hope for this compound to be more successful in the clinical setting. Okay, I think that's all I have.
Quick question.
Yeah.
For the two drugs, are they the indication label as almost the same, or do you think one may have more label expansion?
In this case, AstraZeneca just say it's uncontrolled or treatment-resistant hypertension. I think it doesn't limit it as the for late stage kidney failure patients. So I think the indication for the AstraZeneca is actually broader. If as we remember that there's also a KBP compound that's being acquired by the it's been required and failed in the phase 3 clinical trial. That's for treating the late stage kidney failure hypertension. And the indication for that one is much limited, I think. Actually, the aldosterone surplus or the increase in aldosterone level is quite common. It's much more common than we expected in the hypertension, especially the uncontrolled hypertension. So that means if you use a ACE inhibitor, if you have tried all the blood pressure control medicine, and if you have tried 4 or 5 of them, doesn't work, it's probably due to the aldosterone increase in the kidney. It could be, in some cases, a tumor can cause this, and in some other cases, like late stage kidney failure, the aldosterone regulation is kind of loose and it goes up a lot. So yeah, so it could be a very good anti-hypertension drug, even in common hypertension. Yeah.
Yeah. So you think the better BMPK for the AstraZeneca drug, making it like a best in class?
Yeah, I would vote for this AstraZeneca drug. Yeah, and pharmacologically, it's also more potent. So it takes only 2 milligrams for this to work, and the competitor need to use like 50 milligrams. So it's a better drug.
Anyway, thank you, Haoyan. So let's move on a second.
Yeah. Okay. So yeah, I'm going to switch to the next topic, also relate to the neurological disease. I think this is the second time in the Biospark monthly news we talk about sleep-related disorder. Previously, I forgot which year, Qianbo talked about sleep apnea. It's a very kind of unique sleep disorder, but narcolepsy turned out to be a major sleep disorder. And I think that's the market potential for narcolepsy is really huge. That's why I think it's my pleasure to talk about a very broad, important type of sleep disorder for the first time in Biospark monthly news. And there's several types, major 2 types, narcolepsy type 1 and narcolepsy type 2. There's also something related to narcolepsy. It's called idiopathic hypersomnia, meaning people tend to be more sleepy, sleeping more. Those are the 3 major indications. But the low-hanging fruit is the type 1 narcolepsy. The reason being there is a genetic cause was discovered in 1998, almost serendipitously, because when people were studying the biology about hypothalamus in its regulation, about other neurobiological behavior. So basically, the motivation for Takeda and many other companies to work on narcolepsy is that those patients, the incidence rate is not low, and for those people, especially teenagers affected, the onset age usually happens during the teenage and puberty, and for those patients, they really get a stigma because they tend to fall into sleep in a very awkward way, accidentally, and this will affect their not only at the school performance. For adults, you will have trouble keeping a job because people will view these people as either not very well disciplined, but turn out they have a pathological case going on in the brain to making them having excessive daytime sleepiness. That's why it's a very important disease for a neuroscience-based company try to attack. And another important hallmark for type 1 narcolepsy is what's something called cataplexy. Cataplexy means it's kind of the English word cata means going down, plexy means strike. So it's basically as if those patients were striking down to the ground. So they were all of a sudden losing the muscle tone bilaterally, usually triggered by some strong emotion. If they hear a very funny joke, they will laugh very hardly, but they could all of a sudden falling down. So that could be, in certain situations, could be very dangerous because those sudden drop. But sometimes it's just they will feel some face dropping down, not as severe, but it's just yeah, it's just very awkward socially. And as well as during the nighttime, they also not only they have daytime symptoms, they also have the nighttime symptom. It's that they will paradoxically, although they are sleepy during daytime, but their nighttime sleep is not very well. Usually, it's very fragmented, and they can have some kind of hallucination and a nightmare. So that's really overall a very debilitating disease for those patients. Let's move to the next slide. And the neurobiology of narcolepsy type 1 was having some breakthrough in the late 1990s. If you look at the upper left corner, for people puzzle, what is the meaning of narcolepsy? It's the Greek root. Narco means it's numbness, and the lepsy remind you of the epilepsy, which is kind of like a seizure attack. So it's you can essentially explain the word as if it's a sleep attack, meaning the sleep creeping in wakeness. You're supposed to be awake, and then you have the sleepiness waking in. And neurobiologically, in 1998, people identified a neuropeptide called orexin. It's very interesting that it's a hypothalamus neuropeptide, also serving as a neurotransmitter. But the name orexin giving you the impression that's regulating feeding behavior. But turns out, through another line of research by Stanford researchers, they studied narcolepsy dogs. There's a type of dog that having this narcolepsy phenotype. They do positional cloning. It took them 10 years to find the gene responsible for the narcolepsy in dog. Turned out to be orexin receptor 2. So it's a very interesting convergence of people studying the feeding behavior and the people studying the behavior of narcolepsy eventually converge due to the receptor for the neuropeptide plays an important role in regulating the wakefulness. So turns out there's two different isoforms of orexin, orexin A and B. They come from the same gene because it's a secreted protein called pre-pro-orexin. Pre means there is a signal peptide. Once the signal peptide is cleaved in the cytosol, and you will have these two orexin A and B concatenated into a polypeptide. You need additional protease to cleave them into two different products. Essentially, you can view them as evolutionary. It's a gene duplication. So you have to do post-translational processing to make them into orexin A and B, reminding us of GLP. For people interested, you can watch my GLP-1 webinar in 2024 on Bilibili. And that's essentially very similar. You have cleaving off the pre-signal peptide, then having GLP-1 and GLP-2 in the same polypeptide. Then they have slightly different function in terms of their receptor distribution. It turns out that if we move to the next slide, that there's two types of receptor for those two polypeptides. So interestingly, that for orexin A, also named hypocretin 1, it is not very selective. It can bind to the OX1R, both a GPCR, orexin 1 receptor, OX1R, orexin 2 receptor, OX2R. So orexin A is not very selective. It can bind to the 2 receptor almost equal affinity. Then orexin B is specific, binding to OX2R, highlighting that probably OX2R binding is more central because both ligand need to bind to this same receptor. And turn out to be, yeah, the dog in the dog case, the narcolepsy dog, it's the mutation in OX2R. So that's a very interesting biology. And OX1R turned out to be more associated with addiction and reward-seeking behavior. So that meaning if you want to target sleep-related disorder, usually OX2R might be relatively more important, and but also it won't hurt if you inhibit OX1R and OX2R together if you want to treat insomnia. If you want to induce more sleepiness into this insomnia patient, you may want to antagonize OX1R and OX2R, but OX2R is relatively more important. But if you want to treat narcolepsy, you want to promote wakefulness, you want the agonist for OX2R. And OX1R agonism won't help you. Not only probably won't help you too much because the genetics tell us It's not that much related to narcolepsy. It can even potentially induce addiction. So to treat addiction, you probably want to antagonize OX1R specifically without antagonizing OX2R. So it's a quite sophisticated biology underlying. And then if you look at the upper right corner, the normal sleep threshold, as the y-axis is the sleep pressure, what can you call sleep pressure, meaning as people stay awake for a long time during daytime, by the time of late afternoon or early evening, the sleep pressure will build up. Usually the underlying biology is adenosine. So the reason caffeine can really keep us awake is that caffeine can inhibit adenosine receptor. So that's why it's very interesting. The main underlying substance for sleep pressure is really adenosine. Of course, it has other components as well. So for normal people, they can stay awake for it throughout pretty much whole daytime. Then during nighttime, once the adenosine accumulates to a certain threshold, then the orexin activity will waning off so that people can get to sleep. But you can see those for narcolepsy patients, so reduced sleep threshold, they can fall into sleep even during daytime with those vertical bars called sleeping. So that tells you the central importance of the orexin and orexin receptor mediated signaling in the regulating of sleep. I'll go to the next slide. Then we can see initially the low-hanging fruit for this class of compound is to have antagonist. As June talked about in several previous talk, PD1, PDL1, PD1 blocker, antagonizing PD1 is relatively easier. To make the PD1 agonist is very difficult because there's not too much room to raise the bar. So that's why it's the same story. In this kind of, even in GPCR case, finding an antagonist is much easier than finding an agonist because the Y-type ligand already setting the kind of bar. Your agonist need to be sometimes better than the Y-type ligand orexin. So that's not easy to find an agonist. So they got the low-hanging fruit first. A number of companies like Merck is a pioneer in this antagonist field. I was in Merck West Point when my colleagues were working on orexin receptor antagonist. They did a fantastic job. So they were the first in class. In 2014, they got the first ever drug approved for insomnia called Sovorexant. Then I think Eisai in 2019 got the second in class approved, and the 2022 is a company called Actelion. So the daridorexant turned out to be the best in class, although it's the last approved by FDA. And if you look at the details of each law, it turned out to be the best in class. So those three drugs has already been successful and they approved for insomnia. So antagonists get a relatively smooth path when we're targeting OX1R and 2R. It turns out it's better to antagonize both the 1R and 2R, both receptors. But in terms of treating the disease, any kind of sleep disorder, the left panel just tells you it's a circuit diagram concept. We explain in the dopaminergic case, but in this case, for us to understand the complex neurobiology, it's always a good idea to think about the circuit diagram. The size of the ball means the activity of those nucleus. And one of the key region is called VLPO, is the ventrolateral preoptic region. That region turned out to be important to regulate sleep-wake cycle. When you have very active orexin neuron activity, it can inhibit VLPO so that it will shrink the size of the ball, meaning those activity will be less active so that people will feel less sleepy and stay awake. And then the orexin neuron can also sending a excitatory signal to monoaminergic neurons. Monoamine mainly talking about dopamine, epinephrine, and serotonin. And those typically monoamine neurotransmitter will also keep excitatory case and keeping the wakeful. And then that's why during awake, orexin already playing a central role for people to stay awake. When people need to sleep, healthy people, and then orexin neuron activity will shrink, right? They will reduce. then the VLPO sleep region will really increase the activity. And then the monoaminergic neurons also will reduce in activity, so for people to fall asleep. In the absence of neurons, so narcolepsy type 1 is loss of orexin neuron. The exact cause of the loss has not been fully resolved, but the leading hypothesis is autoimmune. So there's a major leading autoimmune hypothesis because there's HLA type association, but it's not fully resolved. But for autoimmune leading to the loss of orexin neuron, as a result, you will have a reduction of the orexin peptide. It turns out those reception neuron, the receptor not affected. The receptor is still there. That's why this class of drug for agonists can still work if the receptor is still there. It turns out if you without these neuron, then you knock out a intermediate regulator, then VLPA and monoaminergic neurons will directly inhibit each other. That turned out to be a very shaky kind of regulation because it can suddenly switch from awake to REM. People understand REM is rapid eye movement. It's like a different stage of sleep. That stage of sleep, you have dreaming. So typically, when people fall into sleep, you go through non-rapid eye movement and REM. Then you go to multiple episodes of REM. With those narcolepsy patients, in the absence of orexin neuron, those sleep induction can directly from wakefulness to the REM sleep, which is not very desirable because that means it's very painful for those patients. Then if we move to the next slide, I'll tell you how the agonist searching is so difficult. So the medicinal chemists in the field struggle for almost 15 years. Finally, around 2016, Takeda chemists make some major breakthrough. And they, of course, you have to do high throughput compound screening as many other people do, but you have to understand the protein structure. That's why some biologists was able to solve the cryo-EM structure of those two GPCR. And turns out that OX2R, one is inactive, that's the middle gray type of conformation. When you're hitting it with antagonists, some region are closed, some region open up. And then if you, the ideal agonist in the green color, you have to open the region between TM2 and the TM3 have to be open a little wider. Then for that structural biology change to happen, you have to screen a large library compound, and many people was not successful for many years. You can search the patent literature, there's only 2 compounds. Then until 2016, Takeda scientists first getting a hit compound 5. And then that compound, they already get some interesting EC50, but that's still not good enough. It's like 570 nanomolar. It's still not a very good EC50, but then they do some additional modification. Gradually, they, by the time they reach racemic form compound 8, the EC50 for OX2R has been reduced to 140 nanogram. So that's already a good improvement. Then through additional tweaking of the something called, people, medicinal chemists like Hong Yu can tell us the detail. You can further optimizing ligand lipophilicity efficiency, LLE, so that you can trade off between efficacy and blood-brain barrier penetration. So a little bit lower the lipophilicity can making the brain penetration a little bit easier. Then they hit a compound called the TAC925. That's a milestone in the history of OX2R agonist searching. And it's reaching an EC50 of 66 nanomolar. That's very good. And also it's brain penetrant. But that compound, unfortunately, you can only do intravenous injection. It's not orally available. So Takeda chemists has to keep modifying it to increase the oral availability. Then they got this compound TAC994, and they're moving to clinic. Then I know we are short on time. There's a whole story about the unlucky thing about TAC994. The efficacy is very good, like Hong Yu's case with the GSK ADC. It's a very good efficacy. It's very unlucky. It has some, after phase 1, they start to see some mouse models, start to having some idiopathic liver toxicity creeping up. So then in phase 2, unfortunately, 3 out of many patients, only those 3 patients have this idiopathic liver toxicity because some drug metabolite, and you still don't know exactly what's going on, but it's drug metabolite causing the toxicity. They're reaching the threshold of something called Hy's law for those toxicologists. Then they have to, unfortunately, discontinue the compound at phase 2. It's a huge blow to Takeda. But luckily, because phase 1, they already know something potentially happened with liver. They already put more attention to another compound called TAC-861. It's very similar to TAC-994, only adding two fluorine atom and additional CH3 on top of it's making a methyl group in the acyl group. Just these three subtle change, it turned out increase the EC50 and can lower the dose of that compound and taking care of the liver toxicity. So it's a very interesting story. We can have later discussion, but to make a long story short, TAC-861 will only delay 1 or 2 years through 994, smoothly moving to phase 2. And this year, we also got phase 3 results, which is a success. So that's a very good, feel-good story at the end after the struggle with liver toxicity of 994. Moving to the next slide is that we will move very quickly. So far, we only have the phase 2 results published. The phase 3 results will be presented at a future conference, but also Takeda is very adamant compared to their competitor. They want to do twice daily dosing because they believe if you look at the orexin, the neuropeptide kind of concentration, it follows a kind of diurnal kind of profile, meaning there's a diurnal. It's going up and going down. They want the twice daily dosing mimicking that orexin. So the receptor agonism should mimicking the natural ligand. It turned out to be probably helping them with efficacy. Then they designed this phase 2 study, phase 2b study with 112 patients into multiple dosing scheme. But you can see the dosing scheme are mainly twice daily. It's 0.5 milligram twice daily with 3 hours between the two doses and 2 milligram. And then they really holding on this 2 milligram followed by 5 milligram. That turned out to be their best bet from some preclinical data. Then they also have as a control a 7 milligram once daily dose. It turned out to be, you will see that the 2 plus 5 is better than 7 by itself. And there's multiple endpoints. I won't go into too much detail. Then one is the maintenance of wakefulness test, MWT, is just how long you can stay awake when the light turn off. Normal people can be more than 20 minutes, up to 40 minutes. Those narcolepsy patients can only stay awake for about several minutes. That's one of the major endpoints. Then if we move to the next slide, and to give you more detail about the study design of the phase 2, essentially, yeah, it's a very well designed and placebo control and multiple center. You can see the very balanced between the placebo group of 22 patients and the other 4 dose group also almost have the same number of patients. Then move to the final slide next, is that you can see the phase 2 result on the left. And one of the key primary endpoint is the sleep latency. This is the time you can stay awake. Normal people, to be normal, is like more than 20 minutes. If you draw a vertical bar under 20 minutes, you can see the lowest dose did not normalize those patients. Only on average, least square mean is about 12.5 minutes. But once you reach the second dose, 2 milligram, you already have a least square mean change of improvement of 23.5 minutes. And then the 2 plus 5 have the best improvement. It's about 25 minutes improvement. Then the 7 milligram once daily does not do very well. So that tells you that Takeda is definitely moving away from once daily dosing. But Takeda's competitor, Alkermes, still believes their formulation, they can do once daily, but their efficacy data just announced does not look as good as Takeda, and they are falling behind Takeda, but Takeda finished phase 3, and Alkermes phase 3 has yet to be determined when to start. So the stock price for Alkermes has been dropping for the last week. And then you can see some other side effect is, one of the main side effects is insomnia, because when you try to stay awake, of course, you sometimes pay the price of insomnia, but luckily, the insomnia side effect only lasts about 1 week. After 1 week, the insomnia side effect will self-resolve. Then the other side effect is pretty minor, like frequent urination and also more urgency to urinate. So that one probably won't resolve, but that's an acceptable side effect for the drug. So that's why if you want for people interesting to see more detail on the right-hand side of phase 2, you can see that not only the average latency, the wake time, the whole curve is shifting up. Those like ESS score, that's the sleepiness score, the whole curve essentially coming down with the 2+5 dose group best. And also weekly cataplexy rate, that's a very important secondary endpoint. Turn out to be Alchemist is not doing as well as Takeda in phase 3. In Takeda's phase 2, shown here in panel C, you can already see across all those, they have a reduction of cataplexy occurrence. That's a very important indicator, but cataplexy can make people very embarrassed of falling to the ground, all those things. And in Alchemist's case, only the medium dose significantly reduced the cataplexy rate, but Takeda across the board. So overall, then you have this narcolepsy score, NSS also going down in the high dose group. So that turned out to be a very good data set. So Takeda will present the data in Singapore in September early. If the data really have more detail, we will probably share it about Biospark. But they expect to file NDA by the end of March 2026. Overall, they look very confident to be the not only the first in class, probably will be the best in class. Let's see. Let's wait and see. So that's all I want to share. Thank you for your attention.
Well, that's very interesting. So I'm curious, Ethan, you mentioned one of the early program have some liver tox. How did they solve the liver tox with the new generation?
Yeah, so the liver tox, if you move to the compound structure slide, it's a very interesting story. That's the, two fluorine atom was added and making the methyl group a acyl group. That's subtle change. So it's very interesting that the number 861, meaning Takeda found this compound earlier than 994. For whatever reason, they haven't disclosed internally or in publication, they must feel some other DMPK property, although availability may be better for 994. I think Hongyu or medicinal chemist can comment if why these two fluorine and this minor change can basically making the reactive metabolite covalent binding potential somehow get reduced. It's mainly covalent binding for toxicologists in the audience. Covalent binding, but does not, as Shichang always remind me of old story from Merck. Merck paid too much attention to covalent binding, but many FDA-approved drugs have covalent binding, have no liver toxicity. So it's really bad luck for Takeda to have this kind of 3 patient, because this is patient by patient is different. Some patient will have this metabolite leading to covalent binding, and then the ALT, AST just increased by 5-fold. And because this change from 994 to 861, they were able to increase the potency, EC50, then reduce the dose. In the 994 case clinical trial, you often need to go to 90 milligram, even 180. In 861, you can tell you typically 2 milligram, 5 milligram, 2+5. The dose significantly decreased just by making that subtle change of medicinal chemistry. So then it's very clean. Even after phase 3, absolutely no report of liver enzyme increase.
So you think the PK is PK?
Yeah, so Hongyu, could you comment? You are a medicinal chemist. I really
Yeah,
want
so without knowing any information, just my pure speculation, I think that position could be oxidized, and the oxidative intermediate can be trapped by the nitrogen next to it, form a ring called aziridine. So that is chemically reactive. So when you block that with 2-fluoro, so that might block the pathway. But this is a pure speculation. There's absolutely, I don't know anything about this series.
Yeah, for the toxicology aficionado, there's a 2025 Tox Science paper published by Takeda.
Also,
yeah, go ahead, Jun.
to control the necropathy, right? So this has to be brain penetration. So do you believe those two different molecules, their brain penetration capacity is similar or different?
That's also a question for Hongyu. Why Takeda were able to move from 825 to 994, the oral availability, or maybe, Hongyu, could you comment why it LLE optimization works.
Yeah, if I look at the structure, I would say A61 should be more brain penetrable, but when you increase the lipophilicity, it might hit some transporters, efflux transporters. So but if you just look at the lipophilicity, A61 is significantly higher than 994. So it should get into the CNS. If it's a pure diffusion, it should be easier to get into CNS.
Yeah, that's a great point.
But
That means lower dose can be more brain penetrant. That's great.
Yeah.
So Hong, you have any comments? Do you have any comments?
No,
Yeah.
it's just based on nothing else. Just looking at the structure. A61 should be more lipophilic than 984, which should lead to higher CNS penetration.
Yeah. But the toxicity disappear cannot be purely explained, other than Hong, you already commented the reactive metabolites, but it's still very puzzling for toxicologists.
That's very interesting. I also have a related question, regarding neuronal peptide. So this GPCR, right? Neuropeptide, some neuropeptide will deliver signals through some kind of GPCR or some kind of multi-transmembrane proteins receptors. And then I know many people tried to generate small molecules, which is more PKPD or brain penetration, especially for this type of, you have to control, at several hours, right? The cycle, circadian. I think this pathway also has been related to circadian rhythm. So but another way is peptide itself can be druggable as well. But maybe peptide can have some problem for brain penetration, but still can be capable. So why they focus on small molecules? Peptide.
Yeah, that's a very good question. Very good question. Initially, in the early days of this orexin, they have to do peptide mimic, but it turns out the peptide, if you administer peripherally, very difficult to penetrate the brain. It's very, very different from GLP-1. GLP-1, if you remember, is another neuropeptide. It's not neuropeptide, it's a hormone, but it's also a neurotransmitter, but it's produced by the gut. It's the active form of GLP-1 is about 20 amino acid, but because Mother Nature has to select for this to go to the brain, because otherwise GLP-1 have a receptor in the brain, it's produced in the gut. So it has to find a way to go to brain. So Mother Nature makes the GLP-1 brain penetrant. And even Novo Nordisk never expect their good luck, but they just need to increase the half-life. But this peptide, it's produced in the brain by neuron. It never meant to be administered peripherally. That's why they tried the peptidic agonist, could not get brain penetration.
So is that clear? Like GLP-1, for example, how like
it's not clear. Hong Yu, do you have any comments? Or I think I asked that question to Shichang because why GLP-1 somehow can cross the blood-brain barrier. So I don't know. I don't have an answer. Either I want the chemist in the audience to give some answer, but I don't think it's clearly understood.
So people talk about peripheral neuron. Peripheral neuron, some receptors, it can penetrate from gut, neuron to the brain, that kind of access. Peripheral neuron. People talk about that.
Yeah, to me, it's very intriguing to compare GLP-1 to orexin because the way they produce is similar, all the similar post-translational modification, the processing, but it's produced differently. One is in the neuron, the ligand is produced by neuron, and the receptor is inside the CNS. The other one is produced by the gut, but there's a lot of neurons have GLP-1 receptor.
That's very interesting. Anyway, so maybe we can finish the last topic, and then even several minutes to discuss if there is anything else. Hi, Lihua, are you there? Sorry.
Yes, I'm here. No problem. I think maybe let's do this quickly, given I think we're probably a little bit over time. So the reason I want to share this particular publication with this team is less about the individual scientific results of the paper. It's more about, I think, what this initiative, the consortium, and the paper is really indicating in terms of precision medicine in the neurodegeneration diseases. And so as stated by the consortium, problem they are trying to solve, I think we're probably all very familiar with, is in neurodegeneration. There are neither effective diagnostic or treatment. So that's really a big problem as we live longer and with larger aging population. So what I showed here in the diagram, this is a very simple diagram, so there's many nuances, but probably for any disease, if you want to learn from oncology, moving from to where you are today to more precision medicine driven approach, there are fundamentally at least 3 questions you want to really move the needles. So the first one is how to diagnose the disease or how to precisely classify the disease. Because simply calling something Alzheimer's disease is probably many, many different subtypes. So the disease heterogeneity, the different stage of the disease, the different patient may have different prognosis. They all confounding how you're going to treat the patient. So I think, moving from more phenotypically defined heterogeneous disease population to maybe more molecularly defined disease subtype, more homogeneous with the same disease mechanism, will really inviting better diagnosis and better treatment. Then related to that, the question would be, what is the underlying mechanism, the molecular mechanism driving the initiation and the progression of the disease? So moving from we don't know too much, maybe we know a little bit, to by doing large-scale omics molecular study to elucidate the molecular mechanism underneath. And there are some very initial data or insight emerging from this analysis. And the other one, it might be underappreciated, is that if you want to have an effective treatment, you need to be able to measure the disease burden in an objective, sensitive, reproducible way. So if the measurement itself is somewhat subjective, is inaccurate, you can never know what is the treatment effect. So I think I want to address by this particular publication how much they might be started in the beginning to leading toward those directions. And as stated by this consortium, the problem or the vision they want to solve is really improving the molecular understanding, just like we were mentioning here, and to provide a better biomarker and the drug targets. And let's see how much we're moving the needles here. So next slides. And just to clarify, next slides, June. Okay, so I'm only going to talk about one paper. There are 5 publications in this Nature Medicine consortium. I'm only going to talk about the first paper. The first paper is more like the consortium paper, and then the other more specific publications. So just to clarify a few things about the relevance and the significance of this consortium itself is not generating data. Rather, this consortium is really assembling and harmonizing the data that's been generated by multiple institutions and multiple consortiums to bring them together. Obviously, we believe the power of the large data. So the bigger the data set, the more likely you get the better scientific insight. And the other thing is, I think they're really learning from other more open, large kind of data initiative, is that making the data accessible to the community as early as possible before you, as a team generating the data, has done all the scientific mining. So I think they are making this data available using the Alzheimer's disease workbench, not just to the people who are part of the consortium, but also to the community, both academic and industry. So for many of us who came from the precision medicine of oncology era, if you remember when we started the TCGA, which is 2008, during the worst economic downturn from the money of the American CARE Act, that in 2010, only 2 years after initiation, data started to be available to the community, even though the initiative or the TCGA running about 10 years, only finishing in 2018, but the data access to the community started in 2010. So this is also that spirit in terms of a community kind of sharing the data community intelligence to get as much scientific insight out of it as possible. The next slide is going to talk a little bit of what are the data available. So this is a proteomics data set. This doesn't have the genomic data, doesn't have a transcriptome data. It's only the proteomics data with companion clinical data. It's mostly using the SomaScan platform, and it's coming as a collaboration coming from 23 global cohorts and different institutions. And the other thing unique about this consortium versus maybe some other data has been released early on is it's also across different diseases as listed here. So next slide. So at this initial paper, again, this is only the very beginning of mining the data. So it's not necessarily this particular paper giving you too much biomarker or targets for you to act on yet. So they only share 3 scientific results. They call them vignette. So by definition, it's just the very beginning, a small piece of data they are sharing. So the vignette 1 is really 2 pieces of data. One is any bioinformatician will be familiar. You compare your disease versus normal and looking for what are the unique proteomics data or unique proteins that are either up or down regulated in a particular diseases. And one of the interesting data they mined or shared here, I find potentially relevant to the discussion we had in terms of the 3 problems we need to solve is they derive something called a dementia severity signature using just proteomic features. And they correlated that. So this is in the figure IG. And they correlated that with the clinical dementia rating, which is mostly based on the patient's testing score, and showing that it's possible for a molecular signature to really predict the severity of the dementia. And I think this is relevant to the third question we're mentioning. Can you move from something a little bit more, something more subjective, maybe less accurate, to something more objective, more reproducible? So obviously molecular measurement is not subjective, it's objective. So that's a potentially moving us toward that particular direction. And I want to skip vignette 2 because I'm not so sure the relevance of that and given the time. So move 2 slides forward, June, to vignette 3. So vignette 3 is really, they are trying to show the power when you have a large data set, how you can really start to separating the different signals. So in this case, what they are showing is for ApoE4, which is a genotype associated with the early emergence of the Alzheimer's disease. So by this large data set, they were able to separating, as I pointed out in the figure A, a subfigure on the upper left corner, where is a generic profile of the genes associated with ApoE4, regardless whether you have Alzheimer's disease or you're simply a carrier of ApoE4. So those features are just associated as long as you have ApoE4. And then for the G figure on the lower right corner, what they were showing here, the x-axis is the ApoE4 association, and the y-axis is the ApoE3, which is the genotype doesn't have any increased risk to Alzheimer's disease. And then they compare the patient as compare the subject as a E3 carrier with without Alzheimer's disease. And they were correlating the 2 comparison to show there are certain features not only associated with E4 as a genotype, but also associated with Alzheimer's disease. So again, potentially for us to think of, they might be potentially more relevant as a drug targets. So next slide.
So Liu, I do have a question.
Yeah.
So for this association, what does this mean?
Yeah.
That, for example, is a positive AR1. So what does this normally mean? Like means a positive association upon different disease, or like what's
Yeah, I'm losing you. I'm losing you, but I think I asked actually exactly the same question. I don't think I have 100% of the answer. And maybe Xiaobo, if you want to comment on. Xiaobo, are you still online?
Yes, I'm here. This is a great question in terms of what this means. This is a correlation study. It's not a causation study. So that's one thing. The other thing, I think this is a cross-sectional or some longitudinal. I believe this is cross-sectional right now. So this also disease was compared to normal. So we are missing the continuum of the Alzheimer's disease. So I think this is a great start. We still need a lot of work before we can address John's question.
But
Yeah.
generally,
Okay.
Generally, but they have some correlation, like, what 1+, 2+, right? They have some like incremental sunken incremental association. Maybe that is what I talked about.
I read the paper several times. I think the author somehow also didn't go deeper enough because I've been asking this question. Exactly, like if something only associated with the genotype, not with the disease, is that a good thing or bad thing, right? How is it relevant? And the author didn't really answer that question. So next slide. So I think basically, as I said, this is really very beginning. I'm not going to base on this paper to propose which is the biomarker you're going to go test or which is the target you started develop a drug yet. But I think if we're thinking of moving this needle, let's just call that moving the needle. I think from this paper, we started to see using the proteomics data, maybe we're towards understanding the molecular mechanism of the disease. Of course, there need to be functional data. You can't be using some correlation, as Xianbo mentioned, to draw the conclusion of the causation. And the other question is obviously the question of be able to subject or objectively and reproducibly, sensitively measure the disease. Again, as the very beginnings to start to develop molecular signature. Again, if you come in from oncology field, you might be remembering in the early 2000 when there's something called a MemmoPrint or Oncotype DX, which is using gene expression signature at that time, and started to predicting what breast cancer patients are likely to benefit from chemotherapy. And later on, prospective trial was able to validate the utility. So patients do test by it to decide whether they should still taking on additional chemotherapy. So I think started to developing molecular signature is a direction towards something more objective. So to the question of this paper trying to answer, right, kind of developing more maybe sensitive biomarker or relevant drug targets. So are there biomarker candidates from the current analysis? I would say the answer is probably yes, additional work needs to be done. But do we know any one of those genes mentioned in the paper is the drug targets yet? I think there's still some missing links. One is obviously, as Xianbo mentioned, the causation or association doesn't equal to causation. So I think we still haven't completely peeled out those questions yet. The other one is definitely functional information, functional validation. And Xianbo, you want to add additional comments?
Sure, thanks. I was just commenting on we probably need more data. So this is all current existing cohort data. It's not designed to do this in the beginning. Well, maybe is there some planning or thinking of doing this work in the beginning, but not specifically designed for this drug target, for example, to be a fully well-planned, well-designed study. But this is a great start. I think there are a lot of people already start talking about how we do precision neurology with especially for AD, with the continuum of the disease where you have a long time without symptom to the time you start to have a cognitive impairment and to dementia. So that's a long in-between time. And how we properly design and what type of population to include. If I understand the paper correctly, this mostly, they didn't specify, but this mostly from the European dissident with probably better economic status, social economic status than most, not representative of the whole population of AD. So I think this is a great start where if there are more opportunities or more effort to start tracking and to start generating more data, like more omics, with imaging, with other information, with a cross-time longitudinal study. I think that probably give us to better answers to what Li Hua has nicely presented here in terms of the causation, the target, what's the biomarkers.
Yeah, that's great. So I also, I mean, for cancer, we have done this, quite a while, but it's also not very comprehensive. We're just like building the tools step by step. But for neuroscience, neurological disease, tissue, right? Tissue, very hard to get. And then, as we just mentioned about the BBB, for example, if you're talking about small molecules or proteins in the serum or in other places, might not be located, might not reflect what's happening in the brain. So that might have some problem here. And also location, right? Brain is such a delicate, complicated system. So how I mean, I understand, of course, this is in the beginning, how we can solve that type of issues. Like, for example, we talk about
So June, to that question, yeah, I don't know how to solve it yet, but the paper did make an argument of why the fluid proteomics data might be more relevant in these cases compared to in oncology, which is the genotype, right? I mean, it's a somatic mutation. I think their argument is that this is less likely a pure kind of a genetic disease in terms of driven by mutation, but probably more a state disease. So it's kind of the system change. So they argue the protein data could be more relevant.
I see. And also location, right? You can get more different location, more comprehensive, maybe some.
Right. They do have the CSF. They do have the CSF. So for this particular release, that's not the biggest data, but they are collecting the CSF liquid proteomics data too.
So I would rather, in terms of the sampling for the CNS, especially for the brain, there's always an issue because nobody wants to donate their brain samples. But there are some surgeries that well, unfortunately also for CNS neurodegenerative disease, cancer patients typically don't get or don't get AD as much as the non-cancer patient. So you don't have a advantage of taking to when they do the surgery for AD. But there are technologies, if we were talking about brain samples with organelle, with iPSCs, people are starting to do something in that regard to get some handling on the sample. The other thing, the genomics, we are seeing more connections or better understandings of peripheral and central connections. So in terms of different omics, for example, metabolism and lipid and everything. So you start to see a bigger picture of can we use peripheral tissue to get to learn a little bit more about the brain. So I think with a combination of different approaches, maybe we can get a better handle of this question. But this is a great question that has been difficult in the history of studying the brain.
All right, thank you so much, Bo and Lihua. So so Yanhong want to talk about another topic, which is Abivax. Yanhong, do you want to talk about that?
Yanhong. Hi everyone. Ethan, are you still there? Because I saw you made some slides for Abivax. I thought
my slides was from March. From March. It's already been talked about. It's a phase 2B result. This time it's phase 3. No surprise. It's very consistent with phase 2.
Sorry.
Yeah, Ethan, so your voice is in and out. I think I just think that this is kind of very hot topic. Last few days, Abivax announced the positive phase 3 data. Yeah, like Ethan mentioned, it's no surprise. And actually, I was able to dig out back in 2023, they already like the phase 2 result was really good. But just it's very interesting to me as a RNA biologist and have been working on developing therapeutics for RNA for like RNA binding drugs, etc. So when like a microRNA target comes up, it definitely catches my eye. But in this case, it seems that it's not a typical RNA targeting therapeutics that I would be really excited to see. But in this case, actually, the mechanism is actually going after a translation initiation related. So in this case, going after the CBC, which is the cap initiation complex or cap binding complex. So this is a very interesting and very mechanism, and somehow like by reading the news, looks like the small molecule they designed was able to achieve very specific effect on this particular microRNA. And a funny thing about the history of this of the compound Abivax compound or bifazimod. So this obefazimod was initially licensed from Salk Institute, and I think at the time they were actually I think Abivax. You can see from the company name they are actually focusing on antiviral therapy, but somehow this compound turns out it can work on MIR124, which is a microRNA that can regulate inflammation. And they were able to show, so based on the news release back in 2023 from their website, they were able to show that on treatment in patients, they can see really significant upregulation of the microRNA. So I'm reading from here, from 13 to 25-fold, like more than 20-fold in the 50 milligram, 100 milligram group. That sounds like really impressive. And the mechanism they claim, I think this might be, this is the only one I found that they describe very detailed, that the small molecule can actually enhance the selective splicing of the prime microRNA and allow it to eventually generate the MIR124. And also very interesting that this drug, based on the news, the release, they said it does not impact the splicing of other cellular genes. So it's working on the CBC, the cap binding complex, which is known to like working on the translation initiation, mRNA processing, etc. And the CBC actually has two main subunits, CBP20, CBP80, and there are people asking like, what's the MOA of the small molecule and which specific subunit is it working on? People suspect they purposefully did not release the details. For me, from the RNA angle, I think it sounds very interesting that it is known like CBC has a role in splicing, and maybe somehow I don't know how they achieve this kind of specificity of just affecting the microRNA splicing and not affecting other things. So I don't know if the small molecule has additional effects or something we don't know that to allow this kind of specificity to happen. But overall, it definitely sounds very cool and very magic small molecule compound. Okay, that's all what I want to say.
All right, thank you, Xinhong. Any further comments?
Ethan, do you have anything to add?
Not really. It's just, I think the biology already explained in the March. That's why there's nothing new to add. It's just phase 3 data versus phase 2 data. I think it's equally almost as impressive. That's why
and also it's I think for the biotech field, it's kind of I think this last month we see quite some negative news, right? A lot of like FDA negative news from the FDA, but here this is really kind of a positive and exciting news for the field, especially for the overall like stock market, everything kind of being low, and seeing one company doing well and seeing the stock surging so much, it's just kind of good to see for the field.
All right, thank you, Jim. So Ethan, I just checked GLP-1. So I think they mentioned about the mechanism is GLP-1 binds to GLP-1 receptor in the peripheral neuron, which might deliver a signal to the brain, to the stem cell. That's number one. And the second is they might have some BBB penetration advantage. You know, that's people are talking about. That's why this peptide is so effective, and affecting quite.
Yeah, they're also small molecule, but Sean asked a question. I don't have an answer, but it's just some people claim that semaglutide, the Novo Nordisk,
is more peptide-like, has a better safety profile than some of the small molecule. I haven't studied the small molecule GLP-1 agonist too deep to answer that question. That can be another topic we can discuss later. Yeah.
Very interesting. Anyway, thank you so much for everybody for attending and for discussion, for speaking. Yeah, we can stop here and wish you welcome to join our next monthly news. Thank you so much for everything.
Yeah, thank you for attendance.
All right,
Thank you.
Bye.
Okay, thank you. Bye-bye.